Hello, dear friends. Welcome to Histology with Saleh. This is the first video of the series, and I really and sincerely hope that this wouldn't be the last and we would succeed in finishing what we start today and with this video right now. You're most welcome, and for studying histology, we will have a reference book. The reference book that we will use in studying in this series will be Junqueira's Basic Histology. Junqueira's Basic Histology is one of the most famous and successful medical textbooks throughout the world, especially in the field of histology and biology of the tissue. This book is well known, and most of the country's medical systems, the medical educational systems, use this book as their primary reference and textbook in the field of histology and in the field of cell biology. We'll go forward. Histology is a word derived from two other words. The first part, histo, means tissue, and the second part, logi, means to study. So, histology is a branch of the medical science that is used to study the tissue. Easily said and easily put. We'll go forth and look into the tissue and find out what this tissue is that histology is here to study it. Tissue is the part or the component of the body, especially the major component of the organs of the body, which is composed of two components called the cell and the extracellular matrix. These two components, these two structures, form the tissues of the body. We have different types of tissues in the body, and according to the types of the tissues, the types and the specific types of cells and extracellular matrix making up those tissues also differ from one another. Cells in turn are made up of three major parts: the nucleus, the cytoplasm, and finally the membrane. Every cell in the body, no matter what type of cell it is, no matter what shape it has, no matter what function it does, will have a nucleus, will have a cytoplasm, will have a membrane covering this cytoplasm and nucleus. No matter what type of cell it is, in one of the steps of its life, throughout its life, it will have a nucleus, cytoplasm, and membrane. Though some of the cells will, during development, during their lifetime, lose their nucleus, such as the red blood cells or the RBC, which will be discussed later on in the other videos of this series. Extracellular matrix. Like the cells of the body, the extracellular matrix also is composed of other parts. Those other parts that make up the extracellular matrix are the macromolecules. Macromolecules literally mean big molecules. So the big molecules in the extracellular space, or in the space between the adjacent cells, are the molecules that are at the structure of extracellular matrix. These molecules usually form complex structures. These complex structures do their functions according to the place or the position they are, or according to the type they are. One of the most abundant of these extracellular matrix macromolecule is collagen fibrils. Collagen fibrils are the macromolecules of the extracellular matrix throughout the tissues of the body. The second part of this extracellular matrix is the liquid part, or the fluid part. This liquid part is usually also called the interstitial fluid, and is very much similar to the plasma of the blood, and also it is derived from the plasma of the blood. It has the same constitution as the plasma of the blood, and it works, and it is seen by visualization the same as the plasma. Having said that, we'll move on and see what next we have in line. Extracellular matrix has different functions. The most important of which are supportion. This supportion comes to cells. So the cells of an area of a tissue is supported by the extracellular matrix of that same area of the tissue. Next is transportation, and extracellular matrix takes part in transportation. In the function of transportation, or in the act of transportation, three different types of molecules or structures are being transported by the extracellular matrix. First being nutrients, meaning the extracellular matrix takes the nutrients from the blood or from the plasma of the blood and transports these nutrients into the cells, so that the cells can be feeding through these nutrients and can grow using these nutrients and stay alive. So nutrients are being transported by extracellular matrix. Next, the waste products of the cells also are being transported by the extracellular matrix. When the nutrients go into the cells do their metabolism on it, and during this metabolism, some other structures, some other matters become created that are not only not useful for the body, they are also somewhat toxic to the body and need to be excreted from the body. Therefore, the extracellular matrix takes part in this action and takes the waste product from the cells, removes them from the area, puts them into the blood, and through the blood these waste products can go into the systems of the excretory and get out of the body. And lastly, some parts of the body, there are some cells or some specific tissues that the cells of those tissues secrete certain products. These secretory products, or the products that are being secreted, also being transported by the extracellular matrix from the cells or from the glands that produce these products, and after that they are being thrown into the blood, and through blood they could reach their target organs. So this is the total and complete function of the extracellular matrix. But there is also one other thing related to extracellular matrix, and that is their binding. Extracellular matrix do bind to the receptors on the cell membranes. Suppose this is the cell membrane. These cell membranes have these structures called receptors, and the extracellular matrix binds to these receptors, and through these binding and affinity with these receptors connect with the intracellular structures, and thus these actions of transportation and also supportion happens and takes part. Next we have another subject called the preparation of the tissue for being studied histologically. Before that, we need to make and give some introductory lines on these tissues and the preparation of the tissues. Why do we need to prepare tissues, and what is this preparation of the tissues? Since most of the tissues in the body, if not all of them, are thick, and these tissues are non-translucent, meaning light cannot pass through them completely and fully, these structures are not going to be readily visible through microscope, and therefore some procedures need to be done on them so that these structures can be able to be visualized through the usage of microscope. Therefore, microscope is a tool that is somehow near 100% needed to study the tissue through histological studies and histological research, because the cells and also the extracellular matrix that are together forming the tissues are too small to be seen with naked eyes. Therefore, we need the microscope, and we need the assistance of the microscope to visualize these structures and to study them carefully and appropriately. Therefore, when we take a sample from a tissue, doesn't matter what tissue it is, what part of the body that tissue is being taken from. Suppose we're taking this. Okay, thank you. Yes, I know I'm a good painter. Okay, suppose this is the kidney, and whatever problem in it, probably there is a tumor or a mass in it. It needs to be evaluated and studied histologically or histopathologically so that it can be known that this particular mass is either the new plastic, even if it is new plastic, is it benign or is it malignant, or what type of special cells do exist in it, and whether there is one type of cell or more than one type of cell there. This tissue, for example, from this part is being cut through the surgical procedure, and this is taken out. Now we have that tissue in hand in here. Now this tissue, as said before, is thick and non-translucent, so we have to make this particular tissue prepared and ready to be examined through a microscope. Now, I know I'm a terrible painter, but I will try my best to at least make something somehow at least resembling a microscope. Now this is our microscope, and this is the eye of the one who is examining the tissue. For the tissue to be able to travel through this distance and be directly examined by the microscope, it needs to be prepared. Some procedures needs to be done on it, and it needs to be prepared so that it can be easily and successfully examined and studied. This preparation of the tissue for histological examination and for histological study is done through many steps, started from taking the sample or the specimen from the tissue and going all the way to the last part, which is making it on a slide completely ready and without any problem able to be studied. Now this whole process of preparation, depending on the type of tissue, depending on the type of examination that is happening, and depending on the purpose of the examination, could take somehow from 12 hours all the way up to 2.5 days. As said, it depends on the type of the tissue, the type of examination, and the purpose of the examination. Okay, now we will go forth. This preparation, as said, is done through many steps. These steps are those that, okay, I'm sorry, this was a really good painting. Shouldn't have just made it like that. Okay, so 2.5 days. We'll go to the next page. I have something ready there. Yeah. So the whole process of preparation starts from taking the sample, as is shown in this portion, and after taking the sample or cutting by a scalpel, it goes to another process called fixation. Fixation literally means to fix something, to put something in a fixed position so that it doesn't change. And that is exactly what happens in here. The tissue or the cut tissue is put in solutions. These solutions are called fixatives. And these fixatives are solutions that inhibit the degenerative enzymes in the tissue so that the tissue doesn't become degenerated by its own enzymes, and something like autolysis and things like that don't happen out there, and the tissue remains exactly as it has been in the body. So as said, there is fixative that is used in these fixation step. Fixatives are chemical solutions. Fixatives could are different for electron microscopy and light microscopy. If you want to use the light microscopy method and want to study the tissue or the prepared tissue through light microscopy, then the fixative that you are going to use will be usually formalin. Formalin is a solution of formaldehyde with 37% formaldehyde concentration. And if the tissue or the prepared tissue is being studied with electron microscope and through electron microscopy technique, then another form of this fixative is used, which is glutaraldehyde. Glutaraldehyde. Good. Now we will move forward. After the tissue is being fixed and or fixated, it will travel into another step, and this step is the dehydration step. The dehydration step literally means to take the water out of the tissue so that the tissue doesn't have the amounts or the total amount of water that it had when it was taken out of the body and out or cut out of the organ. So this portion or this dehydration part is usually done through a series of alcoholic solution or alcohol solutions, meaning as shown in here, 3 different jars are shown. First it is put into the first one. The first one will usually have a low concentration of alcohol. The second one will have a higher concentration of alcohol. The third one will have higher than that, and the last one usually will have 100% alcohol in it, meaning that when the tissue or the fixated tissue reaches the final stage of the dehydration step, the water or the H2O is taken out of it somehow completely, and no water remains in there, and alcohol takes the place of water. This step is the dehydration step. Next, after the tissue is dehydrated, it will be moved to the other step and other procedures will be done on it. Namely, this procedure or this step is called clearing, and since clearing is a very obvious word with obvious meaning, it is known that in here the tissue is being cleared of alcohol. So the water goes out of the tissue in the dehydration step, and the alcohol that goes into the tissue in the dehydration step goes back outside of the tissue once again in the clearing step. Next, the clear tissue will be put in another jar, let's say, or another dish or another box or holder, whatever you would like to call it, and this process is called infiltration. In the infiltration process, what happens is that the tissue is put in some holder, and this holder is usually going to contain some paraffin, and this paraffin is usually going to be melted type, mean liquid type. So what happens in here, it needs to be put into 52 up to 60 degrees Celsius, so that in this particular range of temperature, melted paraffin could stay melted and also could infiltrate into the tissue. So at the end of the infiltration step, the tissue will be totally paraffin filled. The tissue will have paraffin filling it completely. So the tissue is completely infiltrated by paraffin, or by liquid paraffin, or by melted paraffin at the end of the infiltration stage. Next, the preparation is not finished. The paraffin filled tissue needs to go through another step, and this other step is called embedding. Literally, in the embedding, the tissue is being cut into portions, into slices, and put into another dish or another holder, so that the liquid paraffin that was inside of it is being introduced into another temperature, and this temperature allows the tissue to be completely hardened and form a block of paraffin filled tissue. So this block of paraffin filled tissue, at the end of the embedding step, it is being cut into thin and translucent translucent sections. This process is usually called sectioning, sometimes also called slicing, and this other process, which is sectioning and or slicing, is done on a tool, another equipment. Let me mark it here. Goes into embedding, and after embedding goes to slicing. Slicing is done with a tool or equipment called Microtome. This is the image of a microtome in here, you can see, and this microtome helps the technician that works on it to be able to precisely cut the block of the paraffin infiltrated tissue into slices, thin slices that can be very readily and easily studied by or through a microscope. So this is the end of the preparation of the tissue, but there is one very important thing that needs to be mentioned in here once again, and very properly. The tissue that is embedded and that is cut, in here it differs. If you want to study this tissue through light microscopy, there is one thing that you will do, and if you want to study it through electron microscopy, another thing that you would do. If light microscopy is your goal, then you would put it on a glass slide, and if electron microscopy is going to be your method of study, then you're going to put it on a metal grid. So this is said, and it's still not finished. There's another thing that needs to be done, especially in this light microscopy part. This glass slide that holds the completely prepared part of the tissue or the slide, and also the tissue that's on the other part for the electron microscopy that we talked about, the metal grid, needs to be stained or colored. And why is that? Why do we need to stain it? And how do we need to do it? Is another subject, since we have reached the 30-minute mark, and it's going to be very long if we will discuss all of these processes in this video. We'll take a break right now, cut to the video and stop it, go to the next video for the remaining parts of this lesson. Thank you very, very much for your attention, from the time that you put in watching this video, and hopefully and sincerely hopefully this video will be useful and not just a mere passage of time for you, so that it can help you in your studies, in your medical educations. Don't go anywhere. Be ready for the next part. The next part will continue from the staining portion and hopefully end the chapter 1 of Junqueira's basic histology. Do not forget, dears, do not forget, there's something you need to do. Go to the subscribe button and click it, so this channel can be helpful, useful, and always available for you. Thank you. Have a nice time.